Hello guys and welcome back. This is the part 2 of the microscopy wherein we'll discuss about the microscopes. So basically in this video we are going to discuss about the light microscopes and the electron microscopes. Also we'll discuss about the working of them and which one has the high resolution power. In addition to that, we'll also discuss the type of the electron microscope, that is transmission and scanning electron microscope. Let's get started. So this is the micrograph which you can see over here. Okay, micrographs are what? They are photographs of the cell that are taken using a microscope. Okay, if you have taken using a light microscope, it's a light micrograph. If it's electron microscope, then it's an electron micrograph. So here this picture is of the mustard plant, wherein the different markers has been used to show the stomata present inside the mustard flowering plant. This is the beautiful image and one of my favorite image which has been taken using a microscope. So to appreciate the small beauty around us, microscopes are very important because any teeny tiny things you can observe through the microscopes. Okay, so let's first of all start with the light microscopes. That what is the light microscopes and how they are being used. So light microscopes are the one which is student friendly, because here if you see the picture, most of the students will be familiar with these microscopes. Okay, they are portable, easily used, and they are very convenient in the laboratories. So how they actually work is we'll understand through this ray diagram over here. So the main light source is right here. So here is the light placed. When you switch on the button, the light source will start. First of all, you have the condenser lens over here. It do what? It just gather all the light and they allow them to pass through the specimen. Specimen is placed on the stage, on the slide. So your light source will go, your light will just go and accumulate with the help of the condenser lens onto the specimen. Then here we have the objective lens, wherein if you see there are different magnifications given, 10x, 40x and 100x. That depends on you, that on what magnification you want to observe the specimen. And yeah, the magnification and how the image gets magnified, that we have already discussed in detail in the first part. So if you haven't checked the part 1 of the microscopy, please go and kindly see that video first. You will see that how image will get magnified. Objective lens are of 3 types. Also, what is the use of the oil, that also we have discussed there. So kindly check that. So your objective lens will magnify an image. Then here the intermediate image will be formed. Then using an eyepiece, which is also known as ocular lens, which is generally 10x magnification, you will get your magnified image. If you are watching it at 10x, so 10 into 10, that will give you 100x, 100 times magnified image. So light microscope is simple, no such complications are there. Just there are 3 types of lens: condenser lens, objective lens and the eyepiece lens. Objective lens will help in magnification. Condenser lens will allow all the light to gather at one point, that is on the specimen. Eyepiece lens allows us to observe the image which is formed. Okay, how image is magnified? Please check the part 1. Benefit of the light microscope is that it can be performed on living cells. The advantage of the light microscopes are living cells can be observed. That is, you can observe the normal behaviors of the cell, like when cell is dividing, when cell is migrating. But The main disadvantage of light microscope is it has a low resolution power. So the detailed image is not observed. When you have the low resolution power, remember the resolution is what? It has a ability to distinguish between two objects which are very close to each other. So now if the lens system, they cannot, that is the microscopes, it has a low resolution power, if they cannot distinguish those objects, how we can see the small detailings inside the cell? Okay, so in order to have a high resolving microscopes, electron microscopes has been invented. So we'll discuss this point in detail, that what is the resolution power of the light microscope and what is the resolving power of the electron microscopes, and why electron microscopes are generally used to observe the detail of the cell. So why use electron instead of light? So let's first of all start with the basics. What are electrons? So electrons, they are the subatomic particles that have a negative electric charge, and they are found surrounding the nucleus of an atom known as neutron. So this one right here in between, it's a neutron. Okay, this is the whole atom, and this is the blue ones are the electrons which are surrounding the neutrons. This is what an atom is. So it's a subatomic particle. Now what happens is, when we heat up this atom, that is, we give them an extra energy. So what happens is this: the electrons, they will gain those energy and they will start rotating around the orbits in a very faster manner, and they will escape this orbit like a shooting star. Okay, when we are heating this atom, when we are giving an extra energy, those extra energy will be gained by this electron. They will start rotating in a faster manner in their orbit, and they will leave the orbit like a shooting star, like this. And according to the Lewis de Broglie equation, free electrons behave as a wave. And this electron, it has a high energy. So if higher is the energy, the shorter is the wavelength. And if shorter is the wavelength, greater is the resolution, okay, which we have discussed. So this is how the electrons were used instead of light. They have the higher energy, so higher the energy, shorter the wavelength. So this is what I explained you, that why electrons were used instead of light. So to summarize those 2 points, that why electrons are very suitable form of radiation for microscopes, mainly for 2 major reasons. First of all, the wavelength is extremely short. So shorter the wavelength, the higher the resolution. Part 1 of this video series. So electrons, they have a much shorter wavelength than visible light, and this allows electron microscope to produce higher resolution image than the standard light microscopes. We'll also calculate it. This first point, I'll calculate it, and I'll show you that how they have the low resolution power, the visible light. Okay. Second main reason is, since electrons are negatively charged, they can be focused easily using an electromagnets. Okay, in this magnets, they can alter the beam similar to how the glass lens bend the light. So these are the 2 main reasons why electrons are more suitable. First of all, this one, they have the shorter wavelength so that the resolution power is high. The relation of wavelength and resolution already been discussed. Okay, so see, the first point, I'll tell you in detail that what they mean to say, giving you an example. So this is an electromagnetic spectrum. So where you see the resolving power of light, this visible light, it is limited by its wavelength. That is, it is somewhere around 400 to 700. So visible lights is, the path is around 400 to 700. Now see, general rule says what? To find out the limit of resolution of any of the lens, it is 1/2 of the wavelength used to view the specimen. So microscopes using visible light. Visible light, if you have noticed over here, it is somewhere 400 to 700 nanometer. So if you divide 400 by 2, that will lead you to 200 nanometer. This is the limit of resolution. To know what is limit of resolution, check part 1. So limit of resolution for light microscopes is only 200 nanometer. Okay, and the wavelength of the visible light is 400 nanometer. So what we got is 200. That's the limit of the visible light. That's the maximum resolution obtained with the visible light. So if you see mitochondria whose diameter is 1000 nanometer, this can be viewed under the light microscopes. But the ribosomes, which has the diameter of 25 nanometer only, they are not observed under the light microscope. Why? Because they are not interfering this visible range. They are less than the 200 nanometer. Anything bigger than 200 nanometer easily observed, because the mitochondria, they are interfering the light. Ribosomes, they are not interfering this light rays. They are just passing. Light rays are just passing on from the ribosomes. So in order to observe something which is smaller than 200 nanometer, electron microscopes are used. Why? Because they have the resolution power of 0.1 nanometer. You see, that is way too small. So any detailing can be observed. So that was the first time when electron microscopes were invented. I hope you are clear that why visible light cannot give you a resolved picture and why electron microscopes can. So I hope I am clear with this first one. Wavelength is short, higher the resolution. So next one is what we have, the electron microscopes, wherein beam of electrons are used. Okay, so if you see from the picture itself, they occupy a whole room. They are not generally found in the school laboratories. They are not student friendly. Also, it is the working of the electron microscope is little bit different and complex than that with the light microscopes. It is always connected with a system right here. So generally they are found in the research labs. So let's discuss about the principle of the light microscope through this ray diagram. So first of all, we have the electric gun and an anode which will produce the beam of the electron. So this one is the source of the electron. Then this beam of the electrons will pass down to the condenser electromagnetic lens. The main role of the condenser electromagnetic lens is to direct this electric beam onto the specimen. Next one is the specimen is placed on this grid right here. Now the specimen preparation, it differs for both the transmission and the scattering electron microscope, and it is also very much important. Next one is the electron. After passing through the specimen, it will pass on to the objective electromagnetic lens, which will produce an image. Then the image will pass, then the electrons will pass down to the projector electromagnetic lens, which focuses the magnified image on the screen. So image magnification has been done by the objective lens, Okay, the projector lens's role is what? They just focus that magnified image on the screen or the photographic plate, which shows the image of the specimen, and then later on through this photographic plate we can see it through our eyes. The image is not directly formed into our eyes, wherein the light microscope the image was formed into the eye. Also, if you see, this whole system is placed into a vacuum. This is because the specimen needs to be in the vacuum, because when an electron beam is traveling, and if there will be a lot of air particles, it's going to collide and scatter, and you will never get a clear image. Okay, and therefore the specimens are kept in this vacuum system so that there won't be any hindrance of the air particles. So that's one of the reasons why specimen is dead. There is another reason which is related to the specimen preparation, which we will discuss. So one of the reasons why the samples are dead is because this whole system is placed into the vacuum so that air particles will not collide with the electron beam, and which will not produce a false image. There are mainly two types of electron microscopes, that is scanning electron microscope, which is also referred to as SEM, and the second one is transmission electron microscope, which is also referred as TEM. From the name you might be getting some of the ideas that what is the role of transmission and scanning. So let's discuss in detail first of all about the transmission electron microscopes. So as you see from this diagram, this is the working of the transmission electron microscopes. Okay, this is the simplified ray diagram, wherein you have the electron source right here, the condenser system, which will allow this beam of electron to fall directly onto the specimen. Okay, then objective lens where image gets magnified, the aperture, after that the projective lens where the image will be formed on the screen. So projector lens's role is what? It will focuses the magnified image onto the screen, and then later on the image is viewed through this fluorescent screen. Now basically what happens in this transmission electron microscope is the electrons will get transmitted through the specimen. How they does is, I'll tell you. So if you see this diagram, the electron beam, so imagine this is the electron beam, this right here, and this is our specimen. Because of course specimens are what? They are made up of the molecules, and molecules are made up of what? Atoms. So what happens is, when, say this is the atom of the specimen, okay, right here you have the neutron, and these are the electrons revolving around it. So what happens is, when the beam of the electron, it is passing through this specimen, the beam which is passed through the neutron, okay, those electrons will scatter less. So there will be an electron which will pass through the neutrons. Since neutrons are positively charged, they will scatter less. These electrons, they are scattered less because they are passing through the neutrons. This type of scattering is known as inelastic scattering. That is, these electrons are known as inelastic scattered electrons, where scattering is less. Why? Because they are passing through the neutron, and neutrons are positively charged, so they will attract, they will not allow the more scattering. Now, second type of scatterings are the one where this beam of electrons will pass through the electrons which are revolving around this orbit. So in this case what happens is, this electron will transfers is some of the energy to this electron. which is revolving in the orbit, and this revolving or electrons which are there in this orbits, they will scatter more because they have gained the energy from this electron. So here there is more scattered electron. They are known as elastically scattered electron. Why? Because when this beam of electron, it is passing through the electrons which are revolving in the orbits. So what they does is, while passing, they transfer its energy to this electron, and this electron will gain the energy and they will scatter more. So they are known as elastic scattered electron, where scattering will be more. The image formation is based on this scattering only. How that is possible, I'll tell you. So say this is where your sample is. This is your sample. This is the screen. So now what happens is, this is the electrons which are transmitted. These are the transmitted electrons. So where you see this electron will hit on the screen and you will have the light image. Okay, but where here also electrons will get scattered, no? Because electrons are the beam, so they will scatter in all this direction. So over here the electrons reaching are less. The electrons, they reached very less. Okay, so because of this scattering process only the image formation will take place. I'll tell you. See, imagine this is your sample. Okay, and this is the screen. These are the transmitted electron which will scatter either elastically or inelastically. Now the electron which has scattered more, okay, so they are not in the range, these electrons, they have scattered more. So these electrons, since they have scattered more, the dark image or dark spot will be formed over here, but this electron, they have scattered less. Okay, so here the light image is formed. That is basically you can observe the image formation right here. Remember, transmitted electron, they scatter in two ways, inelastically and elastically. Okay, the one which has scattered more, they will form a dark spot over there, and the one which has scattered less, those electrons will hit the sample and the image will get formed. And this is the reason why black and white image formation takes place in the electron microscopes. So I hope you are clear with the image formation in the transmitted electron microscopes. Now, as I said, the sample preparation plays a major role. So let's see how the sample is prepared for transmission electron microscope. So first of all, what happens is staining is done. Rinsing and staining. So while staining, what happens is they are treated with heavy metals. The treatment requires harsh chemicals and heavy metals, and this heavy metal treatment or harsh chemical treatment will kill the live sample. Why so? Because whenever you are treating it with this harsh chemicals and metals, this chemical, they form an artifacts, and artifacts are what? They are any deviation from the real shape of the image. So they form what? Artifacts are formed because of the heavy metal staining and harsh chemical using. So this also kills the live sample. Okay, so staining, because of the staining and because of the dehydration also, which is the second step. So dehydration and staining will kill the specimen. You cannot observe the live cells anymore. So two main reasons why live cells are not observed. First of all, since this whole system is placed in the vacuum. Second is harsh and heavy metal staining and dehydration. Then next one is the fixation. They are fixed, the samples needs to be fixed with some of the chemical products. After that, they are embedded onto the resin. If you see this grid, so they are allowed to embed on these small holders right here. After that, you have the next one, which is trimming, that is ultra thin section. You need a very thin section of the specimen. Why? Because electrons, they are passing through the specimen. They are cut with the special knife or by an instrument known as electromicroscope. They are very thin slices are cut. So first of all, you have rinsing, staining, and then you have dehydration. Then next one is the trimming of the resin block and ultra thin section. Thin sectioning needs to be done. Okay, then your image, they are fixed with some fixative chemicals like glucaraldehyde, and then they are embedded onto the resin like this. They are fixed and embedded onto this type of grid. Then this grid is placed onto the specimen like this over here. This grid is placed. Have a look at the applications of transmission electron microscopes. Okay, they are used in the topography. Cellular topography can be done, wherein 3D structures of any subcellular macromolecular objects can be done. So topographically, your cells can be viewed. In the cancer research, where ultrastructure of the tumor can be observed, and in the toxicology, to study the impacts of environmental pollution on different levels of the biological organization, transmission electron microscopes are generally used. Advantages and disadvantages are, of course, they have the powerful magnification and resolution, wide range of applications, which we have seen over here. They provide an information on element and compound structures, and high quality and detailed image will be observed using the transmission electron microscopes. But the disadvantages are they are large and very expensive. Sample preparation requires more of the labor works. We have seen the fixation is done, embedding is done, thin sectioning is done. So special training needs to be done. And it has a high maintenance, and the image which are formed, they are black and white. Colorful images are not formed. So these are some of the disadvantages of the transmission electron microscopes. Next one is what we have, SEM, that is scanning electron microscopes. So they scan the specimen and then the image is formed. Okay. So we'll understand the working of the SEM through this image, which is formed away, which is shown right here. Okay, so next one is we have the scanning electron microscopes. Okay, they are generally used to study the surface biology because it scans the specimen. So here, if you see, there are two chambers present. Take a pen. So here there are two chambers present, specimen chamber, and this one right here is column. Okay. High vacuum pump is also present over here, of course, to maintain what? To maintain the vacuum so that air particles will not interact with the specimen, so that we will not come into a picture where clear image is not formed. Okay, so therefore the vacuums are used, which we have already discussed. Then here, if you see, this is the specimen stage right here. Specimen stage can move both the directions, and it can also move up and down. Okay, so this is kind of a revolving specimen, because this beam of electron, it needs to be fall on whole of the specimen. They need to scan this whole specimen. That's why this stage, it moves in all the four directions. Then right here in the column, if you see, you have this V-shaped tungsten wire, okay, which is also known as thermionic cathode. Okay, this V-shaped tungsten wire right here, this one. It is also known as thermionic cathode. So when high electricity is provided here, the electrons get released from this cathode. And down the cathode, you have the another metallic plate, which is known as anode, right here, this one, okay, which has a positive pole, and here is a negative pole. So these both are connected, and because of that, there will be a strong electric field generated, which will accelerate the electron out from the cathode, which is the primary electron, and then this primary electron will fall on the specimen like this. So I hope you are clear how electron beam is generated here. It's thoda different. Yeah, pole. Here the cathode has a negative pole. So when electricity is provided, the electron beam is generated, and remember, the primary electrons, they will pass on from the cathode and move down into the specimen. I'm using the term primary electrons over here because later on you will see that I'll also use the term secondary electrons, which I'll just tell you that what is the primary and secondary electrons. Okay, but the primary source of electron is from here, from the cathode. Okay, next you have the electromagnetic lens right here, this orange one. It allows to focus the beam of the electron onto the specimen. Yeah, so when this primary electrons, it falls on the specimen, specimens are what again? They are made up of molecules, and molecules are made up of what? Atoms. So when the electron beam will fall on the atom right here, this is our atom of the specimen, these are the electrons. Okay, so what happens is, when this primary electron beam, primary source, when they fall on the atom, the electrons which are present in this inner shell right here, this one, okay, they will get knocked out, because of course they will collide with this, and these electrons will knock out. These knocked out electrons are known as secondary electrons. When the primary electrons, that is the main source of electron which is generated through this cathode anode right here, when these electrons will move down to the electron which is revolving in the orbit, they will knock out this electron. These electrons are known as secondary electrons. SEM, that is scanning electron microscope, they use this secondary electrons which has been knocked out from this sample. Yeah, these electrons get knocked out. These knocked out electrons are known as secondary electrons. So then what happens is, the secondary electrons, it will get detected by the electromagnetic deflection, which is connected to SE detector. SE represent secondary electron detector, which is connected to a raster scan generator. So right here, if you see, you have a SE detector, secondary electron detectors, which has been knocked out by the primary electron. They will get detected. Okay, this SE detector, it is connected to the computer right here, and where your image is formed. Yeah, so over here the whole scenario is about the secondary electrons. So you see these blue ones which are coming out from this specimen, they are the secondary electrons. Okay, I hope you are clear with this one. What is the role of the rest scan generator? It just allows to scan the whole specimen. Any of the area should not be missed out because we are dealing with the surface biology. So any surface detailing, if it is missed out, then it will be a big mistake. So raster scan generator, they just help to scan this whole specimen. Okay, they allow to rotate this beam in all the directions on all the surface. Any of the surface should not be missed out. So the area, if you see where electrons are released, you will observe a gray image formed in that area. And if electrons are less released, then there will be a black image formed. Okay, that means if electrons are not knocked out, there the black spot will be there. Where the electrons are knocked out and that electron has been detected through this detector, gray image formation start taking place onto the computer screen. This is the working of the scanning electron microscopes. I hope this will help you. I'll show you the animation of the working of the scanning electron microscopes. See, black and gray images are there. See, so black ones are the part where less electrons are released. Okay, so this is how the scanning electron microscopes works. So this is the overall comparison of the light microscopes and the electron microscopes. This right here picture is of light microscopes. Okay, here. This one is the picture from the transmission electron microscopes, wherein the detailings are also observed. Okay, and this is the picture of the scanning electron microscopes, wherein the surface biology has been observed detail. That is, the surface has been clearly scanned. Okay. So I hope now I am clear with all the three microscopes. Light microscopes, it has a limited resolution, that is only 200 nanometer, and that's the reason why electron microscope was developed. Electron microscopes, since they have the shorter wavelength, so their resolution was high, 0.1 nanometer. And because of that resolution power, only the electron microscopes allows us to give the more subcellular, that is the detailing structure, and subcellular compartments can also be observed, which you can see right here. So again, electron microscopes were of two types, transmission and scanning. Transmissions are the one which uses the transmitted electron. The electrons will pass through the specimen, and that's the reason why the specimen needs to be thin. In scanning electron microscopes, there is the role of the secondary electrons, which has been knocked out by the primary electrons. These electrons will get detected and that will be focused on the monitor. The main difference is electron microscopes, they cannot visualize any of the live processes happening because the sample is dead. Two reasons. First is they are placed in the vacuum. Whole system works under the vacuum. And second is because the sample preparation has a heavy metal, staining and harsh chemical requirements, they kills the live cells. So sample preparation will be the second main reason why the live images are not formed, where electron microscopes live images are not formed, okay, in the electron microscopes. So you can remember two reasons. Apart from that, electron microscopes, they are expensive, require more labor work. Okay, you have just seen the photographs and everything, high maintenance is required. So generally the students, they use the light microscopes, and this is enough at the school level. So in the next video, that is part 3, we will discuss about the measuring of the cells. There are 3 main techniques which we'll discuss in the detail. So stay tuned, and if you have liked this video, do subscribe my channel and click the like button. Okay guys, thank you so much.